All right, so I'm going to be mainly focusing on pulmonary hypertension on this presentation. So, to kind of talk about some of the molecular mechanisms of the vascular remodeling that we see in pulmonary arterial hypertension. So we all kind of know that pulmonary hypertension is very devastating, and it's precipitated by hypertrophic pulmonary vascular remodeling of distal arterioles to increase pulmonary arterial pressure and pulmonary vascular resistance in the absence of left heart, uh, lung parenchymal or thromboembolic disease. And despite our medical advances, we see pulmonary hypertension all the time, and it leads to right ventricular dysfunction, failure, and early death. The studies I found talk about cellular and molecular mechanisms of pulmonary artery remodeling to find pathways for intervention. There is a heritable genetic component to pulmonary hypertension, and there's also evidence of other genetic perturbations like pulmonary vascular cell DNA damage, activation of the DNA damage response, and variations in microRNA expression. They all play a part in dysregulation of proliferation and apoptosis signaling pathways, akin to what is observed in cancer, and changes in cellular metabolism, metabolic flux, and mitochondrial function, and endothelial to mesenchymal transition as key signaling pathways that promote the pulmonary vascular remodeling. So the pathological findings that characterize pulmonary hypertension are hypertrophic distal pulmonary artery remodeling, inflammation, fibrosis, and thrombosis, and neovascularization. In some vessels, this process leads to obstruction of the vessel lumen and create complex vascular lesions that are known as plexiform lesions and are pathogenomic for pulmonary hypertension. And this obviously increases PVR and PAP and causes hemodynamic load on the right ventricle. The right ventricle goes through remodeling to compensate for the increased hemodynamic stress, but is prone to failure leading to premature death. Now to kind of talk about a little bit of the DNA damage and pulmonary hypertension and the DNA damage response. There's been evidence of DNA damage and somatic genetic abnormalities in pulmonary vascular cells isolated from patients with PAH, pulmonary arterial hypertension, which and it was actually shown in endothelial cells from plexiform lesions that were shown to have microsatellite instability. In the hypertension, endothelial cells also showed cytogenetic abnormalities. DNA isolated from explained pulmonary hypertension as compared to explained disease control and non-disease control lungs found mosaic chromosome abnormalities in the hypertensive lung. Once a hypertensive patient had a chromosomal deletion of BMPR2, which is a genetic cause of pulmonary hypertension, and somatic loss of chromosome 13, which has SMAD8, which means that DNA damage in hypertensive lungs happen at a higher than expected rate. I also read that DNA damage predates the onset of pulmonary hypertension and is likely an intrinsic property of cells in individuals that are susceptible to the disease. They found changes in about 30% of pulmonary artery endothelial cells isolated from explant lungs as compared to only about 5% in cells isolated from donor lungs. The pulmonary artery endothelial cells with evidence of chromosomal abnormalities and circulating peripheral blood mononuclear cells also had more DNA damage, assessed by measuring chromosome breakage and loss. DNA damage in the endothelial cells are correlated with reactive oxygen species production by the endothelial cells. The DNA damage response is activated in pulmonary arteries isolated from patients with hypertension, and pulmonary artery smooth muscle cells showed evidence of DNA damage. And there's activation of the polymerase I, which is part of the DNA damage response and contributes to DNA repair by binding to strand breaks in DNA and generating poly at the break site. Activation of the PRP1 increases cell survival and proliferation through a mechanism that involves MIR204 mediated activation of nuclear factor of activated T cells, C2 and hypoxia-inducible factor 1 alpha. In the presence of DNA damage and pulmonary hypertension is also associated with downregulation of BMPR2, which in turn affects the DNA damage response by regulating expression of the DNA repair gene, breast cancer 1. Downregulation of BMPR2 is a hallmark of pulmonary hypertension and is associated with a drop in the DNA damage response that further compromises the genomic integrity of the cells. So now to kind of talk about a little bit about the microRNAs regulating the gene expression in hypertensive patients, or pulmonary hypertensive patients. So overall, they have a pretty big role in modulating gene expression. MIRs are a group of small non-coding RNAs of about 22 nucleotides that silence gene expression by binding to the by binding to the 3 untranslated regions of mRNAs and inhibiting translation or promoting degradation of the mRNAs. Several studies performed global surveys of MIR expression in order to determine the profile of differentially expressed MIRs in pulmonary hypertension. One study studied about 337 miRNAs and identified 6 that were upregulated and only 1 that was downregulated in explanted pulmonary hypertensive lungs as compared to controlled donor lungs. Another study by microarray using plasma from 8 patients with pulmonary hypertension compared to 8 healthy subjects. This study identified differences in 58 MIRs between the groups with the MIR 150 highlighted as the most significantly reduced MIR in pulmonary hypertension patient plasma. And then examination of MIR expression in remodeled pulmonary arteries from patients with severe pulmonary hypertension identified MIR 126 and MIR 21 levels as being upregulated in plexiform compared to concentric lesions, while expression of MIR 143, 145, and MIR 204 were higher in concentric lesions. And the role of several MIRs, including MIR 1719, 143, 145, 204, 214, 21, and 131, 301, and regulating pulmonary hypertension relevant signaling pathways. And the disease phenotype has been confirmed in experimental models of the disease. So to kind of briefly go over the changes in cellular metabolism and mitochondrial function, which a lot of us learn in our biology classes. So to kind of quickly review that, cellular metabolism can change quickly to respond to environment. Glycolysis occurs in the cell cytosol and generates pyruvate that's transported to the mitochondria, where it serves a substrate for pyruvate dehydrogenase as part of glucose oxidation. When pyruvate dehydrogenase is inhibited, there can be uncoupling of glycolysis from glucose oxidation, which would lead to increase in the intracellular lactate levels. The mitochondria is where fatty acid beta oxidation yields ATP, and acetyl coenzyme A for entry into the citric acid cycle. And there's a crosstalk between glycolysis and fatty acid oxidation, such that a change in the activity of one pathway influences the activity of the other. And this is shown by the Randall cycle, where a reciprocal relationship was discovered between glucose oxidation and fatty acid oxidation. And the aerobic glycolysis is a shift in metabolism to glycolysis, followed by the fermentation of lactic acid instead of pyruvate oxidation in the mitochondria. And the net gain is to produce ATP to meet energy requirements and to promote cell growth and survival. The shift to aerobic glycolysis usually occurs when a key mitochondrial metabolic enzyme like pyruvate dehydrogenase or pyruvate dehydrogenase kinase is inhibited. The mitochondrial function is also reliant upon the structure integrity. So within cells, mitochondria continuously divide and join together in processes referred to as a fission and fusion. And the mitochondria are pretty important for oxygen sensing, and this is dependent in part on the stability of the mitochondrial network. Metabolism is perturbed in pulmonary arterial hypertension with consequences for cell proliferation, survival, and apoptosis. In pulmonary hypertension, the pulmonary vascular cells show a rise in aerobic glycolysis as a result of normoxic upregulation of HIF-1 alpha and inhibition pyruvate dehydrogenase, the pseudohypoxic state leads to normal oxic activation of HIF-1 alpha. Occurs due to changes in redox state. And the cellular redox state is affected by decreases in levels of mitochondrial reactive oxygen species generation that inhibits oxidative metabolism. Mitochondria and pulmonary hypertension are fragmented due to activation of fission regulator dynamin related protein 1, have decreased expression of both complex 1 and superoxide dismutase, and have a hyperpolarized membrane. Mitochondrial superoxide dismutase can be repressed transcriptionally via methylation of 2 CpG islands in the SOD2 gene. And interestingly, this phenomenon occurs in the lung vessels only, and it's not observed in the systemic circulation. Normoxic activation of HIF-1 alpha in pulmonary arterial or hypertensive upregulates pyruvate dehydrogenase kinase isoforms 1 and 2, which leads to phosphorylation inhibition of pyruvate dehydrogenase with a switch to aerobic glycolysis. And the molecule dichloroacetate is potential therapy in experimental pulmonary hypertension, and it helps to improve mitochondrial structural integrity and function, decreases pulmonary artery smooth muscle cell proliferation, and regresses established pulmonary hypertension. Then I also wanted to kind of talk about a little bit about the endothelial to mesenchymal transition. And I read that endothelial to mesenchymal transition really happens in pulmonary artery hypertension, and that these transformed endothelial cells are the sources of the alpha smooth muscle actin positive cells that accumulate in the medial wall and participate in hypertrophic remodeling. And it's where endothelial cells show phenotypic plasticity and acquire properties of the myofibroblasts or mesenchymal cells. This process is associated with loss of tight gap functions, dissociation from the basement membrane, and migration into the medial layer. During this process, the cells lose their typical endothelial markers such as CD31 and vascular endothelial cadherin and start to express the alpha smooth muscle actin and vimentin. It's stimulated by members of the TGF beta that signal through the kinetoconal and non-kinetoconal pathways, and also can occur after exposure to inflammatory molecules such as interleukin 1 beta and tumor necrosis factor alpha, or angiotensin II receptor type 1 activation. And in patients with pulmonary artery hypertension, examination of pulmonary artery plexiform lesions revealed that luminal endothelial cells were swollen, and some cells expressed both endothelial markers and alpha smooth muscle actin, a finding not observed in controls. And the presence of was also confirmed in pulmonary hypertensive lungs by showing expression of mesenchymal genes, including the fibronectin and cadherin and vimentin and the endo-MT related transcription factor twist. To kind of go over another study that I found that talked about synthesizing of bile acids of pulmonary hypertension, and it really kind of went over how there's really little knowledge or research done on the metabolic heterogeneity of pulmonary hypertension. So they used a variety of high throughput mass spectrometry, and they found bile acid metabolites, which are normally products of the liver and gallbladder, were increased in pulmonary hypertension lungs, and profiling the heterogeneity of the pulmonary arterial hypertensive lung, they found a new pathogenesis for it. And in their evidence, the literature suggests that the metabolic pathway abnormalities characterize and may play a significant role in the development and progression of hypertension. So, for example, pulmonary artery endothelial cells, or I'm going to kind of refer to them as PAECs, and pulmonary hypertension share similar hyperproliferative characteristics as malignant tumor transformation that's accompanied by significant metabolic shifts to support the anabolic growth and energy metabolism. Also, mitochondrial oxidative phosphorylation with absorption of glucose happens in pulmonary artery endothelial cells development, and there is a rise in the hemoglobin with the hypertensive sample group without diabetes, which pretty much means that there's an impairment of glucose homeostasis and pulmonary hypertension. And another thing this article found was vascular changes with chronic hypoxic patients, and they saw a disproportion between the glycolysis, glucose oxidation, and fatty acid oxidation. But pulmonary artery endothelial cell culture with disturbance of bone morphogenic protein receptor 2 gene actually had a lot of metabolic changes. And when glycolysis is disturbed, it amplifies the TCA cycle and fatty acid metabolites with reformed pathways of oxidation and end up leaving the hypertensive lung, which tells us that pulmonary hypertension has vascular remodeling and hypertension. So many metabolic pathways are reprogrammed during the remodeling. Heterogeny could have a big part in ATP energy supply and molecular pathogenesis. In this article, they found a rise in bile acid metabolites and pulmonary hypertension lung tissue that's linked to bile acid synthesis related transcripts. In other words, means that the synthesis of bile acids could contribute to the pathogenesis of pulmonary hypertension. So as a fun fact, taurocholate is a product of conjunction of cholic acid with taurine. Its sodium salt is the chief ingredient of the bile, and it acts as a detergent to solubilize fats for absorption. It's used as a cholagogue or chlorectic, which are GI agents that stimulate the flow of bile into the duodenum or stimulate the production of bile by the liver. And in this picture right here, we have mass spectrophotometry fragmentation of taurocholate in a control group in hypertensive lung. The top part shows a negative ion, selected ion chromatogram for taurocholate, and the compound identification depended on the fragmentation spectrum corresponding to the taurocholate standard. The top and bottom parts of the figure were run separately, and there was clearly a very small peak in the normal lung. And then as far as what kind of materials and ways that they did this, I'm not really sure if it's too relevant, but I figured I'd share it anyway because I thought it was kind of interesting. There were biochemicals that were found in lung tissue, and they compared 8 normal and 8 pulmonary hypertensive patients. And the hypertensive patients were all end stage and all had lung transplants. The lung samples were acquired during the lung transplantation for the control group. They got their samples from normal tissue of the cancer patients who had lobectomy, and biospecimens and data were composed with written consent and approved by the IRB. Unbiased profiling using liquid gas chromatography coupled to mass spectrometry was done, like I mentioned earlier. As far as transcriptomic analysis, the mRNA samples from the normal and hypertensive lungs were obviously separated, and bile acid-related profiles were compared between both of them. The RNA analysis was done by triazole extraction, and biotinylated cRNA was done via Affymetrix protocol. After the fragmentation, cRNA were hybridized on gene-chip genome array, and then they were scanned. The data was analyzed with the Partag Genomics Suite 6.6 using the Affymetrix default analysis settings, and the definition was set up using the Genomics Suite 6.6, and the genes that changed a lot were decided by the t-test with a false discovery rate of 2-fold. As far as immunoblotting, protein concentrations were figured out by using the BCA protein assay. Equal amounts of the protein lysates were separated by the SDS page and transferred onto nitrocellulose membranes. The membranes were incubated for overnight at 4 degrees Celsius with the following antibodies from Abcam-R, anti-CYP7B1. And after wash with TBS tween, the blots were incubated for 60 minutes at room temp with horseradish peroxide conjugated antibodies, respectively anti-rabbit antibody signals from immune reactive bands were visualized by fluorography using ECL reagent, and the intensity of individual bands and immunoblots were quantified using the NIH image program. So immunohistochemistry, so the sections of both hypertensive and normal lung tissue were fixed for 4 hours at room temp with PBS made of 4% formaldehyde, permealized for 30 minutes in Trident X100, and incubated with 5% non-fat skim milk and PBS for 90 minutes. Sections were incubated for 180 minutes at room temp with antibodies for anti-CYP7B1, and the sections were incubated with biotinylated secondary antibody and visualized with a dab. And stained cells sections were visualized with Zeiss LSM510 confocal microscope. So here's another MS fragmentation spectrum of glycolate in normal and hypertensive lung. So a negative ion is selected ion chromatogram for glycolate in both studies, and the identification of glycolate compound depend on MS fragmentation spectrum, and they were both obviously ran separately. For this figure, the hypertensive lung had a distinctive bile acid pathway. Intermediates in the pathway showed higher levels of glycine and taurine, conjugated bile acid in the hypertensive lung. The green boxes showed the normal lung data, and the pink are the hypertensive data. Quantities are in random areas explicit to the interior standards for each quantified metabolite and protein concentration. This figure was done, this is microarray data, showed that there was a lot of expression in the hypertensive lung with a gene encoding cytochrome P450, family 7, subfamily B, and polypeptide 1. Western blot analysis of CYP7 beta 1. Expression in both study groups. Lung lysate was filled up and blotted with antibody against CYP7B1 and GAPDH. There was a rise in the CYP7 beta 1 expression in hypertensive study, and the enzyme protein was also higher. And the CYP7 beta 1 positive immunostaining in new formed small blood vessels, arrows, in the plexiform lesions of occluded pulmonary small vessel and pulmonary hypertensive lung. Representative micrographs of immunostaining in the pulmonary hypertensive lung sections are shown with the anti-CYP7 beta 1 in the pulmonary vascular endothelial cells. So overall, this study really wanted to figure out the metabolic mark of pulmonary hypertension, and pulmonary hypertensive lung had higher levels of bile acid metabolites, including the primary bile acids, taurocholate and glycocolate, quinodioxylate, and glycodenocoloxylate. And biodiol acids are typically made in the liver and gallbladder from cholesterol by 7-alpha hydroxylase, known as cytochrome P450. That's an enzyme that limits the rate of the synthesis of bile acid, and microarray analysis helped discover that cytochrome P450 beta 1 was a lot higher in the hypertensive lung. And the more analysis they did, they found that CYP7 beta 1 enzyme was higher in hypertensive lung, which means the higher bile and acid metabolates may not be caused by reflux from the esophagus, but it might be actually coming from the lung, which obviously leads us to conclude that hypertensive lungs have the capability for synthesizing bile acids, and a potential biomarker could be a rise in bile acid. But overall, thanks for listening, and these are my references.